I'm Lacey Berger. I'm the moderator for today, and we are new to the Dupe 15Q Alliance. We've been with you guys for just a few months, and so we have already had our eyes opened and have already learned from our presenter today that our child had been misdiagnosed, which is nice that we have that information now and can move forward with it. So without further ado, I want to go ahead and introduce our speaker. It is Brenda Finucane, and Brenda is a licensed genetic counselor and the associate director and professor at the Autism and Developmental Medicine Institute of Geisner Health System in Lewisburg, Pennsylvania. Her clinical and research activities have focused on the genetic causes of neurodevelopmental disorders, including Dupe 15Q syndrome. Ms. Finucane is widely published and has been in leadership roles in the professional and advocacy organizations throughout her career, including a term as president of the National Society of Genetic Counselors. Ms. Finucane was the co-founder of IDEAS, Isocentric Exchange, Advocacy, and Support, which subsequently became the Dupe 15Q Alliance. So please welcome Brenda Finucane.
Thanks, Lacey. Woo. It's wobbly up here a little bit. So welcome. I imagine many of you are kind of first-timers or have a first time at a conference at least. And I will say, as a genetic counselor, if you're confused about the region and the different terms that get used for Dupe 15Q, you should well be. There are many other genetic disorders that are far simpler in terms of their genetics. This is a very complex region of the genome, and there are a lot of different ways that you can end up with Dupe 15Q syndrome, and so if you're confused, you're in good company. It is one of the toughest regions even for genetic counselors, and for genetic counselors, I'll often get calls, saying, wait, run this by me again, how can this happen? So it's just a lot going on in this region, and it's a complex condition. Before I start, just a couple of disclosures. I work for the Geisinger Health System, which is out in central Pennsylvania. I'm a consultant for the National Fragile X Foundation and for LabCorp and for Ovid Therapeutics, which is interested in targeted therapeutics for neurodevelopmental disorders. I'm going to talk about Dupe 15Q syndrome, walk you through quite a bit of the vocabulary that we use so that you're kind of all on the same page, show you what the region looks like, and when we talk about an isodicentric, what we mean, etc. I want to mention that a geneticist who was one of our originals as well with the Dupe 15 Alliance. Her name is Carolyn Shannon. She's coming in shortly, and she and I will also, at the end, be just doing some quick reviews of lab reports that people have submitted with questions. So she'll be joining me for that after this talk. So a syndrome, let's just start from the basics. What is a syndrome? A syndrome is a pattern. It's a pattern of physical and behavioral characteristics. So when we think about Down syndrome, which most people in the general population know about, even people who aren't involved in science or anything like that say, well, yeah, when I see somebody with Down syndrome, there are certain features that they have in common that makes me know or recognize that that person has Down syndrome. That doesn't mean people with Down syndrome all are exactly the same. They have wide variability just like the rest of people in population, but there's just something about the pattern of features that you see that's instantly recognizable. So that's a physical pattern. There are also behavioral characteristics that can be a pattern, such as autism, as I talked about yesterday. There's a number of issues with social interaction, et cetera. So that's autism in a way is a pattern of behaviors. And so when we talk about syndromes, we're saying, each time we see the Dupe 15Q syndrome, there's a particular pattern that we can recognize. Now, that being said, it doesn't mean every person with Dupe 15Q syndrome is exactly the same. There's wide variability in the way kids look or adults look or the way they behave or how they function intellectually. But it's still enough overlap, and I'm speaking to the choir here. You're all here at the conference. We can see the similarities, not just in behavior, but in looks, even though we can recognize and appreciate how different people are as well. So Dupe 15Q syndrome has come over the years by a number of different names. This is actually a condition that was first described many decades ago when we had just chromosome studies to look at. And the reason this was one of the first ones picked up is because in many cases you could see, even on those old chromosome studies, that there was an extra piece of genetic material, an extra chromosome. Okay, so there are many much newer genetic diagnoses that weren't able to be even seen, but this one was picked up even with old technology. So what are the features that go along with the Dup15q syndrome? Well, typically there's infantile hypotonia, or very poor muscle tone, a baby who's very floppy at birth, and that often is something that triggers an evaluation in a hospital. There are some subtle facial differences, but really not anything that anybody in the general population or even trained geneticists would necessarily pick up on. Now, obviously, you know, I would bet parents here at this conference, you'll say, yeah, I would recognize it if I saw it sometimes, and that's true. But I can tell you, and in your experiences, you probably know that many clinical geneticists just don't see that many kids with Dup15q, so they don't really pick up on that kind of look, that shared look that they all often have, like they're siblings to each other. Intellectual disability is very frequent in Dup15q, but it's widely variable. And epilepsy can occur in just over half of people with the condition, especially in babies who may have, in the newborn period or just at birth, infantile spasms. Autism seems to occur in a majority of people, and autism, again, is just a group of behaviors that involve social interaction differences and communication problems. I did put sudden unexplained death in a minority. I'm sure those of you who have been on the Dup15 website and have read anything, we know that there's a small, very small percentage, but it's a real percentage of individuals who may pass in their sleep, and it's unexplained. The majority of those individuals do have epilepsy, and so this is a phenomenon not just in Dup15q, actually any condition that involves epilepsy. You find this sudden unexpected death in a very small percentage. So this is not just unique to Dup15q. In fact, the condition is called SUDEP, sudden unexpected death in epilepsy, of whatever cause. And it just so happens that Dup15 has seizures as one of the pieces of it. So it's not anything that special probably about Dup15. It's more that anything that has an epilepsy characteristic can have this small increased risk. And Dup15 syndrome, by definition, includes a duplication of what's called the PWACR, or the Prader-Willi-Angelman critical region, and I'll walk you through what that means. There are a number of other names for Dup15q syndrome. In the past, this was called inverted Dup15q, and it's also referred to as an isodicentric 15q. This is a funny story. When I first started what is now the Dup15q Alliance in 1994 with a parent who had a son with this condition. At the time, it was called inverted Dup15q, so we called our group IDEAS, Inverted Duplication Exchange Advocacy and Support, as an acronym. And a few years later, the genetics community threw out inverted Dup15q. They didn't like inverted Dup, but they called it isodicentric. And we had just purchased new letterhead. Thank goodness, isodicentric still fit with IDEAS, so we kept it there, right? So I was like breathing a sigh of relief that they didn't change it to something completely different. Sometimes it's called partial trisomy 15, sometimes tetrasomy 15q, interstitial Dup15q, etc. So I'm going to start basic, but I won't get into too many details, but just to orient everybody, when we're talking about Dup15q syndrome, we're talking about a chromosomal disorder, something that is affecting a chromosome 15 in this case. And chromosomes are the packages that our genes come in. So every single body cell has a number of chromosomes, and they really are the packages that contain the genes, and the genes are made up of a chemical called DNA. So you hear all these terms sort of bandied about, but genes are basically stretches of a chemical called DNA. Deoxyribonucleic acid. And they're all bound up together into these packages that we call chromosomes. And, you know, for a long time, we were able to look at chromosomes. That's why Dup15q has been known for many decades, actually, because some of the earlier technologies were just the ability to look at the packages. We could look and see these relatively large structures called chromosomes that are in the cell, Even with just a light microscope, you know, we could take somebody's cells and look at them. And this has been around since 1959. We had the first ability to see chromosomes. So if you looked in somebody's cells, every cell in the nucleus of the cell would have these chromosomes scattered about in this way. And then in the laboratory, years ago when we were looking at chromosomes, they would actually take a picture of these and cut out individual chromosomes and then line them up in size order. They're not neatly lined up like this in your cell, unless you're really rigid or something. I don't know. Mine are probably like really lined up. But control freak, all my chromosomes have to be lined up. Sorry. But what we know is that, apples, Max. Okay. Can you see? You can't see my cursor here, but they're lined up not only in size order, but also according to their stripe patterns, or their bands, as they say. So number 1, you can see that they're the tallest chromosomes, and they have a certain stripe pattern. And it's very different from the number 6s, for example, which are shorter and have a different kind of a stripe pattern, etc. Those ones that are kind of twisted, that doesn't mean there's anything wrong with those chromosomes. It's just when the picture was taken, they're in 3 dimensions, right? So they're just bent over or curled or whatever. This is a perfectly normal chromosome pattern. So in most people, what you see is that there are 23 pairs. The first 22 pairs are the same for males and females. The 23rd pair are the sex chromosomes. And in females, you have 2 of those X chromosomes that are down there, and they don't have any Y chromosome. Oops. So there's the sex chromosomes, and that's a female. And in males, there are typically 46 chromosomes. There's 1 X and 1 Y chromosome. The Y chromosome actually is only found in males and has a small number of genes that are really specific to male characteristics, such as, you know, sitting in recliners and watching sports and those sorts of things. Sorry, can't help but go there when I talk about it. So what were we looking at when we saw these chromosomes? Well, basically, and this is old technology, we really don't do chromosome studies much anymore at all because we have much better technology. If you can imagine, there's about 20,000 genes packed into these chromosomes. So there's lots of genes. Now we have the ability with microarray technology and whole exome sequencing to actually go at the level of the gene. So really to get into those packages and see the code that's going on. We couldn't see that in this. But with Dup15, because it was an extra chromosome, it was kind of an easy one to pick up back in the day. So what were we looking for when we did chromosome studies? Always looking for, are there extra chromosomes, missing chromosomes, like dramatically shorter chromosomes, so missing sections or extra sections. And the most common thing that one would find on a chromosome study is an extra 21. So in this case, this is a male. He's got an X and a Y, and he's got an extra 21, so 3 rather than 2 chromosomes. And in that case, we see somebody who has Down syndrome. In Down syndrome, they always have an extra number 21. When we're talking about Dup15Q, we're talking about the number 15 chromosome. I forgot to mention, the reason they come in pairs is we get one from mom and one from dad at the moment of conception. And we really do get half of our genetic material typically from each parent. Sometimes it's hard to believe that. I have 2 children. They look nothing like me. They look exactly like their father. If I hadn't been awake when I gave birth, I would have thought, but I'm in there. I know I am. Half of me is in it. Sometimes it's personality traits, blood type, you know, other types of things that we have. So what you see on the outside is not the sum total of genes. Those are just genes that determine our physical characteristics. We really do get half of all of our genetics from each parent. So if I was to take that chromosome 15 and draw it out like a cartoon, this is what it looks like. And I want to define some words for you that we talk about when we think about chromosomes. And first of all, I mentioned bands, so those are the stripes. And these bands become apparent in the old days when we did chromosomes. When they put certain stains on the chromosomes, some of the parts of the chromosomes would soak up some of the stain and not others, and so it would create these bands. And this was a good way for geneticists in the laboratory to be able to all be on the same page. They knew that this was a 15 because there's a certain pattern to those bands. A centromere is this little pinched-in place up near the top here on 15, and every chromosome has one. In some chromosomes, it's more in the middle, and the 15, it tends to be up near the top, and it's really kind of like a pinched-in area of the chromosome, like a waist or something like that. And that's important, well, because the centromere is important for when the cells divide, but it also kind of defines our nomenclature, or how we name things. Because when chromosomes were first visualized in the 50s in France, what they saw was, well, here's this little pinched-in piece, this centromere, and there was usually a small part of the chromosome above that and a bigger part below it. And so they called the small part the P arm, because it's petit, and they call the long arm the Q arm, because Q follows P in the alphabet. Nothing scientific about that. It's just P and Q. And so when we talk about dup 15Q, we're talking about the long arm. That's why it's Q. This thing up at the top is called a satellite. It looks like a satellite. It's like hovering above the chromosome, and it's attached there. That'll become important when I talk about the inverted duplication. So a couple of other things that you might read about. CNVs. There's a lot of talk these days about CNVs. In fact, there are thousands of CNVs that are known. These are copy number variants, basically deletions or duplications. A difference in the amount of genetic material. At the most extreme end, one could say that, trisomy 21 or an extra 21 chromosome in Down syndrome is a CNV. It's an extra copy of the whole chromosome. But even very tiny little microdeletions, little tiny sections or duplications are also CNVs. So CNVs are deletions or missing sections of genetic material, or duplications, extra material. And then some CNVs, and this is important because many of you, I think, have had chromosomal microarray testing. That's kind of the standard now for a lot of kids with developmental delay. And sometimes the report might come back and say your child has dup 15Q, but it says, oh, and by the way, we also found a VUS, which is a variant of unknown significance. This happens actually quite often. And these VUSs are the bane of every genetic counselor's existence. And it's the price we pay for great technology that allows us to pick up variation, but the lab is way ahead of our ability to interpret what they mean. And so there are these things we pick up and say, that's interesting. It looks like a little bit of extra material, but we have no idea whether it means anything or is just sort of a trait. So we find a lot of VUSs. And, I think it's an okay price to pay, because back when we were just doing chromosomes, our diagnostic yield in a population of kids with developmental disorders was only about 3%, which meant, yeah, we could tell them yes or no, we found something, but 97% of the time we never found an answer. Our diagnostic yield right now with microarray is 15 to 20% of kids with developmental disorders. You can find a specific abnormality, but we have a lot more VUSs. That's okay. I'll pay that price to get the 20%. And as we learn more about these VUSs, and for those of you who've had them come up on your reports, the geneticists often say, please come back in a year. Maybe we'll know more about it. Maybe we'll know, oh, this is really benign. We've seen this in a lot of people. You don't have to worry about it anymore. Oh, we actually found this, and we think it might be related to some other feature that your child has. The words microdeletion and microduplication get used sometimes, and it's the same thing. It's just that we're talking about something that can't be detected visually on a chromosome. So these microdeletions, if you looked at somebody's chromosomes, like the pictures I just showed you, you wouldn't be able to see them. They're much too small. Everything looks fine. But if you go with the new technologies of microarray, et cetera, you're able to detect them. And mosaicism means that there are two or more different genetic patterns in the same individual. Typically, when we find something like a dup 15Q, an extra piece of chromosome 15 in an individual, it's in every cell of the body. And the reason it's in every cell is because when that sperm and egg came together at conception, that was the first cell. And then all the cells multiplying after that still carried on that extra material. So we expect that every cell of the body is going to pretty much have the same complement of genetic material. However, sometimes that dup 15Q condition happens a little bit after the first cell. So the conception was fine, the cells divided a little bit, and then there was some error that caused a dup15q to arise, and then all the cells after that had dup15q, but some original ones still just had the regular complement. And then you would find two different populations of cells in the person, and that's what we call mosaicism. And think about a mosaic on the wall. It's different populations, different glasses to make the whole picture. That's where that term comes from. So mosaicism is typically detected, it can be suspected on a chromosome abnormality, and they'll find different populations. The microarray won't always pick it up, but the FISH should be able to pick it up in a lot of cells. And we do know that, it's not uncommon for any of these chromosomal disorders to have mosaicism, and in dup15q we definitely see it every now and then, and you may get reports of different populations of cells. So if we're talking about isodicentric, let's say here's our chromosomes again. The first 22 pairs there, and this is a female. She's got 2 X's. That's the 23rd pair. But if you look at her number 15s here, she has 2 typical 15s, one from mom and dad, and here's that extra one. We know it's a 15 because the bands kind of match up for the top of the 15, the 15P, and then some extra material around the 15Q. So back in the day, for those of you who have adult sons or daughters, let's say, you may have been diagnosed just on a chromosome study. So they could pick it up and they say, boy, it's not an extra 21 like Down syndrome. This looks like a 15, and that's all we knew about it. And then there was a technology called FISH, which stands for fluorescence in situ hybridization, which is a type of chromosome study where they could actually add a tracking probe, what they would call a probe. So a piece of genetic material known to be part of the 15q region we're interested in, and they would attach to it a fluorescent tag or a dye so that when it found its match, it could light up. And so what they would do in this case, and it doesn't show up really well here, but it would go to chromosome 15 because it came from 15, and if it found its match, it would light up. And in people with 2 typical chromosome 15s, there would be 2 bright lights. And then if you had a dup15, there would be a 3rd or a 4th because that extra chromosome sometimes had 2 sections of 15. So FISH was used pretty extensively in the late 90s through the 2000s, and now when people get a chromosomal microarray test, they often will then do a FISH test because this helps us locate, is it separate or is it within the same chromosome? So FISH is still important, and FISH isn't just for dup15q. There's FISH for all kinds of things. Every chromosome you could make a FISH test for it, but it's very specific to what you're looking for.
Is FISH where you find out whether the extra chromosome is from the maternal or maternal side?
No, the FISH is not. The FISH only locates, like, is it interstitial or is it extra, isodicentric, which I'll talk about. There's another test called MLPA, which is methylation, and they typically don't do that for this. We know that these duplications are primarily, and especially when they're extra, are primarily maternal in origin. The only time we would do it is if there was an interstitial duplication within a chromosome, and we might want to know, is it paternal or maternal? But that's a test called MLPA. So a chromosomal microarray, so this is kind of state of the art these days. A child comes in who has a developmental problem, or maybe that hypotonia, poor muscle tone in the newborn period, and or epilepsy, for example, early on. They're trying to figure out what this is. So microarray is a very, very powerful technology because it's not just visually trying to look, is there a little piece missing or whatever. It's actually taking chopped up genetic material and fusing it on an artificial substance, like a barcoded slide, for example. And DNA is interesting in that when you split it up, it's 2 strands, right? And if you separate those strands, they'll find each other and go back together again, right? So what they do is they separate out the strands of DNA and they chop it up. And then on this slide, they have typical DNA with the amount of genetic material we expect to have. And then they put the child's or the person's DNA on top of it, and then it gravitates and tries to find its match. And if it finds too much of its match, it'll say, boy, there's too much going on here, which is what we would say in duplication 15q. If it was not finding its match, you would also be able to detect a deletion. So microarrays detect copy number variants. Deletions are duplications, and thousands of different copy number variants. So boy, it's like the mother of all genetic tests. It's amazing. You don't even have to be looking for it. So in the past, if you wanted to think about Dup15q, you know, you might have to think about it, do a chromosome study and say, oh, is it really, you know, what is this chromosome we found? Is it really 15? In this case, we don't have to be looking for anything. We'll just do the test, and 15 to 20% of the time in individuals who have developmental disabilities, we find a specific diagnosis that tells us this is it. This is what's causing the problem. It's very, very powerful. So chromosome 15 is an important chromosome, and again, to make your lives even more difficult, it has to be one of the most complicated chromosomes out there because it's got this region that we're talking about that is a little bit different depending on whether it comes from mom or dad. Most other chromosomal regions, it doesn't matter whether you inherited it from mom or dad, but this is what we call an imprinted region. So the properties in the dad's 15 that he passes on are going to be different than the properties in the mom's 15 that she passes on. And there are only a couple of regions in the whole genome that are like that, and welcome to difficult genetics. It happens to be this region. So not only do you have imprinting, differences in whether you got it from mom or dad, but then there's also, was it inherited as a whole extra chromosome, or is it plugged in or embedded into an existing chromosome, and how big is it? So there's all these different variables we have to deal with. So deletions of genes within this region here, as you can see in red, at the very top of the Q arm, right underneath the centromere. This is what we call the Prader-Willi and Angelman critical region, because when you delete it, when you're missing sections, if you're missing the 15 you got from your mom in this region, you get Angelman syndrome. If you're missing that section in the chromosome that you got from your dad, you have Prader-Willi syndrome. So I'm saying, you know, these genes, when you're missing them, it can give you two very different disorders. We're talking here about duplications, so getting too much overexpression of genes, and that's where we get Dup15q syndrome. So the smallest deleted region that we know that can cause Prader-Willi or Angelman is called the PWACR. And core duplications, and many thanks to Guy Calvert for kind of coming up with these, this wording that has stuck over the years. I remember the funny phone calls we used to have about cores and edges, but it's actually really true. So there are a lot of different things that can happen in this region. And as I'll show you, you can have people who have a deletion in this region, but it doesn't include this PWACR. They don't have Angelman or Prader-Willi syndrome. They don't have, if they're duplicated, but it doesn't include this critical region, they don't have Dup15q syndrome. They have something completely different. And you know, for a while there, some of those families were coming to these meetings, and it was very confusing. I think we've sorted it out. So we at the Dup15q Alliance, and it's kind of stuck a bit, is come up with the idea of core duplications. These are duplications that include this region, and these cause the syndrome with that pattern of physical, medical, and behavioral findings that I mentioned. Then there are these edge duplications. Duplications, again, around the same region, but they don't include the Prader-Willi Angelman region, and these are different Dup15s, but they're not part of the syndrome. And so they really have very different characteristics. They have some overlap, but they really are not the same condition at all. So that's why it's really important to know your lab result. Are you a core or are you an edge? And when you come to the Alliance, you may know people ask, you know, can you send us your lab report? And it's really important. We don't want you spending time with the wrong group. There are other groups, Facebook pages, whatever, for edge duplications. And they're different. They really are different disorders. If you're in this group, hopefully you've all got the core duplication, so we all are talking on the same wavelength. And so the lab report is key. Lab reports are really, really important to review. And right on cue, Carolyn Shannon just walked in. She's going to be with me up here at the end, and we'll be looking at some lab reports that some of you sent ahead of time, or if you brought them with us. And so you can get different types of lab reports. You could have a karyotype, that's the old kind of study, also called a chromosome study, and then you have a result that looks like this, 47 chromosomes. Sometimes they say IDIC 15Q11. Sometimes some of those reports say PSU, which is a pseudo-dicentric chromosome, same kind of thing. So there's different ways that you could write this down. Importantly, some people's reports say they have 46 chromosomes, the usual number, and that's a first clue for us that maybe this isn't a whole extra chromosome. Maybe this is an interstitial, which is less common, but it's a piece, an extra piece of the dup15, but it's embedded into one of the existing chromosomes. In fish, your tip-off here is the word, or the letters ish after it. So that means it's a fish study. So a fish study will typically have ish in there, and it'll tell you what genes were in there that they were looking for. And so you see that SNRPN and D157Z1. Those are things that they would give that fluorescent probe to, and they would go search, and they'd light up if you had more than the usual. And then microarray is your ones, it's kind of the standard of care now. We really don't do chromosome testing anymore, but now microarrays that'll look for thousands of different deletions and duplications. You know it's a microarray when it says ARR in the beginning, because that's an array, and then it has all these coordinates about what's missing. But an array can tell you something's extra or missing, but it can't tell you where it is. So when you find a dup15 extra of this material, typically the recommendation is go back to fish and go back to that earlier technology and say, okay, but where is it? Is it hanging out on a whole extra chromosome, or is it embedded within one chromosome, an interstitial? Yeah, this gets complicated. You know, it's like, I go to other genetics meetings, and this talk takes about 2 minutes. Okay, it's deleted, it's not inherited, whatever. This is real, I feel your pain. This is a complicated region. So let me talk a little bit about paternal and maternal. These are some great slides that, I guess, Carolyn, you were at UCLA back in the early days when they did these graphics for some of our first, but they're really pretty slides that show the chromosomes. And so I mentioned before, when you saw that chromosome picture, they come in pairs because we get one chromosome from dad, one from mom at that moment of conception, typically, right? And so there's a paternal copy and a maternal copy. So here they're green and purple. And so when we talk about dup15, here are the various things that can happen and that we need to consider. Well, we have to know, is it a whole extra chromosome, an isodicentric as we call it, or is it interstitial? Is it embedded within one chromosome? Is it de novo, meaning it started out of the blue just in the child, was never in the family before? That's typical of isodicentrics. Or is it familial? The interstitials can sometimes be familial. How many copies are there? Is it one extra copy, 2 extra copies, 3 extra copies, 4 extra copies? And it can be variable within even the same person. They may get 2 extra copies of one section of the dupe, and then they may have 3 extra copies of a whole other section. What are the breakpoints? That means, how big is it? Like, what does it involve? What is this extra piece? What are the sections that are at either end that tell us how big this is? Who was the parent of origin? Did it come from mom, or did it come from dad? And typically with isodicentric, or the extra 15 dupe, what we know is that typically to have symptoms, it comes from mom, but there can be paternal or maternal interstitial duplications. The maternals are found much more commonly. And then finally, that question of mosaicism. Is it in every single cell, or is it in just some cells? So there's all these things we have to think about. It's not just, yes, you have it, no, you don't. There's a lot of variability. So if I was to take that chromosome 15 and drop it down on its side, as you see here, lengthways across the top, in the red box there, that's our 15Q11 to 13 region. And you need to understand, too, it's not 15Q11 to 13, and if you really want to impress your geneticists or doctors next time you go in and correct them if necessary, it is 11 and it is 13. And the reason for that is because it's chromosome 15, Q is the arm, and 11 means it's in band 1, and then there's subbands and subbands of subbands. So it's not 11. There is no band 11, but there is a band 1, and there's a subband 1. There's a band 1, and there's a subband 2, right? So it's 11 to 13. So here we are. That's the region of interest. And if we were to take that red box and blow it up like this to show it, you'll see underneath there that it's got, and this is a really nice slide again, thanks to Carolyn, just sort of showing the breakpoint 1, breakpoint 2, breakpoint 3, 4, and 5. So some people can have a very small duplication that just involves between breakpoint 1 and breakpoint 2, but it doesn't involve what we call the Prader-Willi-Angelman critical region. They don't have this syndrome. They may have a duplication of this different area, but it's not our area. Here's our area, okay? This is the PWACR. It's between breakpoint 2 and breakpoint 3. It contains a lot of genes in there. We know about a lot of those genes. We're learning more about those genes. We know what happens sometimes when you don't have enough product from those genes, and we're learning things that happen when you have too much from those genes. You may have heard about some of the research involving animal models of Dup15q duplication, and so the animal models are basically mice, flies, other animal models in which this condition has been bred into the mice on their chromosomes that are related to this region. And so in an Angelman mouse, for example, you would have underexpression of one of the key genes in this Prader-Willi Angelman region, UBE3A, and there's also a Dup15 mouse, and it has overexpression, like it's got too much of that product. So by having these animal models, it really helps us to, hopefully in the future, understand where we can go with treatment. So that's the PWACR. So let's talk first about isodicentrics. And isodicentrics have many names, as I mentioned, IDIC 15, inverted Dup 15. Sometimes, years ago, they were called supernumerary marker chromosomes, and you still see that on some lab reports. And basically what that supernumerary means is you usually have 46 chromosomes. This is an extra chromosome. That's all that means. And they called it a marker because back in the day, they weren't sure where it was coming from. Like, what's it made up of? What's the material in this chromosome? So sometimes you still actually hear that or see that on some reports. A bisatellited supernumerary, sometimes these are called, meaning this extra chromosome seemed to have two of those satellites on the end of it. And those are just the top part of on top of the P there that are just above the centromere. And so those are important for cell division, and so they sometimes said, well, it's bisatellited, and so you may hear things about satellites. Sometimes it's called tetrasomy 15, tetra meaning 4, meaning in certain situations there are 4 copies instead of 2 copies of the PWACR, and then sometimes it's called partial trisomy 15. In other words, there's 2 typical 15s, and then there's a partial piece of 15 that's in there, so it's a partial trisomy. That's really kind of inexact when you think about it. If you had a whole other 15, which is not compatible with life, you'd have trisomy 15, but there is no trisomy 15, so they called it partial trisomy 15. So here's what it might look like. And for many of you looking in your lab reports, if we were to draw cartoons of what your kids' labs look like, you'd have 2 15s, and then you'd have this isodicentric, an extra chromosome, and it's made up of that top section kind of flipped over, duplicated and flipped over, and then stuck together again. Sometimes we call that, as I said, a bisatellited chromosome. You can see that there's satellites on both ends, so it's bisatellited, or sometimes just one satellite. Duplication of all of the P arm and part of the Q arm. So typically with this, remember that centromere, that squeezed in waist there? That P arm above that is wholly duplicated, but really the thing that's causing the symptoms is not the P arm on 15. What's causing the symptoms is those extra genes in Q. And so we've got part of the Q that's duplicated. And if you have 2 normal 15s and then you have one of these, then you've got 1, 2, 3, 4 tetrasomy. You've got 4 copies. Right. Well, not the other 2. They've only come from 1. So the 2 are there and they're intact, moms and dads. And then somewhere in early egg development, when mom's eggs were being formed, there was breakage and reconnecting, so you ended up with what's called a derivative, this little chromosome, and then she passed on her normal 15 and this little derivative, and then dad passed on his normal 15. So it's not something that happens typically after the conception, it was just something that was in one of the eggs, and these things kind of happen pretty routinely, but this is an area that happens fairly commonly when it comes with genetic diseases. So
3 copies or 4 copies?
I'll show you in another slide, but imagine you have 2 15s. You got one from mom and one from dad. And then you got this extra at the moment of conception, isodicentric. And you can see here that if you've got a P from the mom and a P from the dad, and now you have 2 Ps here, that's 4 Ps. If you have part of the Q from mom, part of the Q from dad, now you've got 2 extra Qs here, that's 4s. Mm-hmm. Yeah. So here we are. Here's the 4. You see up here, thanks, that's a better slide to show it. See at the top there, you've got your P arm from mom, P arm from dad, and then you have this extra chromosome that's got 2 more P arms, so you've got 4. Same thing. Now, not everybody has exactly the same size of a duplication, so some people may not get a whole extra copy of all of the Q genes, so they may actually only have 3 copies versus 4. I'll show you a little kind of crude graphic in a little bit that might help show this in action as well. So when it's called when it includes the PWACR, that's when it causes the syndrome. If it doesn't have the PWACR, you're not talking about the syndrome. You're talking about a different disorder. Important. And these are of maternal origin. Isocentric 15s have been known for decades. They are of maternal origin. If a dad's sperm, somehow this breaks off and forms one of these, we really don't see people, I mean, there may have been some, possible papers reporting something like this, but, it's really not something we see paternally inherited. This is a maternal phenomenon. And that maternal phenomenon speaks to the imprinting as well, that, we know that if there's a deletion and you get the mom's chromosome deleted versus the dad's, they cause 2 different disorders. We know for the duplication, what we see when there are symptoms is the mom's isodicentric will cause those symptoms. And they're de novo. So this is important. De novo means out of the blue. Nothing you did, neither parent has it. I know, and we have for decades said, parents should have studies done to see if it's in there, and they don't turn out that the parents have this. It's de novo. It comes out of the blue every time. And there's a bit of debate in the healthcare community now in genetic counseling. We're trying to save healthcare dollars. Should we still be recommending that we do parental studies when we find an isodicentric? They're not in the parents. Should we be testing those parents? I don't know. As a genetic counselor, parents are anxious, and when they want to be tested, it's some form of reassurance, I think. But, if we're thinking about healthcare dollars, really, if we know this is not something that recurs, should we be doing it? And I got into quite a debate with my boss, David Ledbetter, who's one of the pioneers in medical genetics over time, and I was in favor of let's just test the parents anyway. It gives them that reassurance. They never have to ask it again, to which he gave me the namby-pamby award. And anybody who knows me, I am not namby-pamby, but he said I was very namby-pamby. I just need to bite the bullet. We shouldn't be testing parents. Sorry, I digress. And sometimes these are mosaic. So sometimes you have cells that don't have the extra, and sometimes you do. All right, so here's my crudely done graphic here. So let's show how this happens, and this might happen in some cells. Let's say this is the region we're talking about, the P and then some of the Q, and then imagine that gets duplicated, and imagine that gets flipped on its head and stuck back on. There's your isodicentric, right? But this doesn't happen as an ongoing process after birth. This is already there. This is happening in eggs, so that you might have a woman and she has this constellation just accidentally in a couple of her eggs by accident. And if that goes on to produce a child when her eggs actually divide and she gets her own maternal chromosome and brings that along, that's where you can conceive a child with DUP15. So the other type of DUP15 origin, which is less common, the most common one we see is that, isodicentrics, is the interstitial DUP15Q. And in this case, there are 46 chromosomes. Typically, it's not 47. There's no extra chromosome. It's embedded within it. So you can see the chromosome here on the right is larger, taller than the one on the left. They're both 15s. And back in the day when we could only visualize chromosomes, they'd say, this one looks bigger. It might have something extra in there, and took a bit of detective work to figure this out. But you'll also see that that band is quite a bit bigger. So there's a duplication in there. We now have techniques, since fish, et cetera, that we're able to clearly show there's extra material in there. So there's no extra chromosome here. It's often inherited. So when you find an interstitial, it's always important to check the parents to see if they have it and to see if it came from one or the other. It can be maternal or paternal. The vast majority of interstitial DUP15s that we know about are maternally inherited, but thanks to Dr. Larry Ryder, who's part of our professional advisory board, he's actually been very interested over the years in those paternally inherited ones, which are associated with a different kind of behavioral characteristics, ADHD, not so much intellectual disability, but maybe some some autism sometimes, some more behavioral overlay. But again, that's less common than the maternal interstitial. In the past, these were often missed on chromosome studies. You know, when I said in the past when we did chromosome studies, we had about 3% yield, and 97% of the time we couldn't identify a cause. Well, now we have very powerful technologies. So often they were so small, you just couldn't see them. They can be detected by FISH very easily and microarray. And again, critically, when the dupe includes the PWACR, then you have dupe 15q syndrome. If it doesn't include the PWACR, you may have a duplication of part of the region, but it gives you another disorder or a different condition. So imagine this, that you have interstitial dupe 15, and when eggs and sperm, in this case, are forming, okay, let's say there's that little piece of the region we're talking about, and rather than forming a whole other chromosome, what we find is that there's actually a duplication of that region, and if that goes on to conceive a child, then you would have a child with dupe 15q syndrome. So there's the moms on the left and the dads on the right, and you can see just even size-wise, one's bigger than the other, but with very small interstitial dupes, there may not be that much of a size differential. You can't count on your eyeballs to pick these up. You really need FISH or some other technology. So some of the most common types of IDIC 15 chromosomes, again, back to that extra chromosome, would be where you'd have a very symmetrical one, where you'd have a breakpoint 3 to breakpoint 3 connection there. And so you can see there's 1, 2 copies there, and then you have your 2 regular 15 chromosomes, again, 4 copies. On the one on the right side, there's a difference. It's asymmetrical, and some of you who have sent your reports over that we went over this morning, your kids have asymmetrical dupe 15s, so it's not that uncommon, meaning one of the sides has 1, 2, 3, 4. It goes up to that section of the chromosome. The other one is larger. It's a larger duplication, and so one side has more of the material than others, the other side. Let me show you this graphic. So for the breakpoint 3, breakpoint 4, these are regions of the chromosomes. So here, this pink region here, okay, is what we're talking about in these breakpoint 3, breakpoint 3. And let's say this pink region, this is that chromosomal region, goes up to breakpoint 3. And then for this type of chromosome, what you've got is that you've got 2 pieces of chromosomes that stick together. Each of them has a duplication of that region. On this side, what you see is that, yes, you've got a duplication of that pink region again, but there's also on the top, it goes a little bit longer down to breakpoint 5, and so what you've got is a little bit more genetic material there, but it only came from 1 chromosome. So what you might have is tetrasomy, 4 copies of the pink region, because we're thinking about you still have 2 normal 15s plus this, and you might have only trisomy for this little region, okay? It's really complicated. Here's the bottom line. There's a lot of variability in this region. Even just the chromosome studies we went over today, a lot of differences among the kids. But it doesn't really translate to anything we could hang our hats on in terms of differences in the way the kids present. Okay? We can't say that time we see a kid who's verbal, who has dupe 15q. Oh, in that case, we always see this pattern. It doesn't work like that at all. You can have a kid who's very verbal, who has a relatively large, symmetric dupe 15q tetrasomy of this whole region, and you can find somebody else with the exact same region. They're nonverbal and have more cognitive challenges, more autism, etc. The one thing we can say is that as a group, individuals who have that interstitial, not the whole extra chromosome, but that little embedded piece, as a group, they tend to be higher functioning. They tend to have fewer of the cognitive challenges. They tend to have autism for sure, and autism is often, very variable. But you can't always say that. I've met people with interstitial duplications who have far more challenges than maybe somebody with a full dupe 15 from an isodicentric. So we can't really make prognosis on this. So I would not, after explaining all this to you, don't get too bogged down on what are my breakpoints and what's the prognosis going to be. We can't predict prognosis from breakpoints. We don't have a crystal ball.
I recall when I was at the Orlando conference, I don't know if it was your computer or your sisters, when we shared our breakpoints, they had like a pie, and they said, look, your breakpoints, you had already collected enough data, I guess, from the alliance to say, you fall in this group of families in the alliance have your breakpoints. And you were able to pinpoint that.
We are. So that was and the comment was, I think it was the Orlando conference. We actually had, with the Alliance, reviewed all the hundreds of lab reports over the years that came into the Alliance that families have submitted, and I remember it was 386, I think, lab reports that we reviewed. First of all, we were wowed by the variability. It was amazing how many different variations there were of Dup15q that included the PWACR. We also found that there were some people who did not have Dup15q syndrome, and we needed to route them to another support organization. They didn't have the PWACR. But it was very variable, and we had created some graphics then, they're not being shown at this conference, but like from that group, how many people had isodicentric? It was the vast majority of people in the Alliance. How many people had isodicentrics that were asymmetric? How many were more symmetrical? So there's wide variability, but again, it doesn't predict prognosis, unfortunately. It would be helpful if it did. Okay, so here's the PWACR again. Here's that region. And there are some important genes in here that we always want to be thinking about, and there's work going on with those various genes. Important things to think about. The prevalence in clinical samples. Now that we can do microarray, and laboratories out there are doing thousands and thousands of microarrays, laboratories have been looking at their data. And data's been published on over 30,000 microarray results. Now, keep in mind, microarray, most people in the general population don't ever have a microarray test, right? So it's a really biased sample. It's a sample of people who have symptoms. They have clinical symptoms. Usually, they can sort it out and say, of all the people who have autism whose samples were submitted because they had autism, Dup15q turns out to be by far the most common copy number variant. So it's attracted a lot of interest in the autism world because it is something really that we can point to. About 1 to 3% of all autism is due to Dup15q. And about 1 in every 500 of those microarray samples will have a Dup15q. So it is fairly common, and it speaks to the growth that we have in the Alliance over the years. A lot of work went into getting this group going and resuscitating it over the years, trying to get enough families. There's a lot of energy to put into growing a group like this, and I'm just so proud of the group, and the families have taken it forward, and we're so big now. We have 1,400 families involved, or people involved in our Alliance, and it's going to be growing all the time because microarrays and other tests get done all the time. There is a gene within that region, GABAB3, which is among the most common findings in epilepsy when you have a problem with that particular gene. So that gene alone, even in people who don't have Dup15q or deletions of the whole region, if they have a problem just with that one gene, very often it's associated with epilepsy. So we know that's a very important gene for understanding the seizures that happen in kids with Dup15. So that's a good thing. When we start to know these individual genes, what do they do? How can we treat things? We're far ahead. I can tell you, and I said this yesterday when I was speaking, it may not feel like it to some of you parents who are like, living with the diagnosis and learning about it for the first time and still sort of sorting this out, but you're so much luckier than individuals who really, the diagnosis is based on autism or some behavioral diagnosis that's far removed from the cause. When you know a cause, we're really close to understanding what some of those genes are doing. What does this mean? Can this help us with better treatments, et cetera? So we're really in a much better place knowing that, yes, it might be autism, but we are many steps ahead of that. We know what's causing it in the sense that we know it's coming from this region.
What does it mean, like a person who doesn't have a deletion or duplication? What does it mean when you say they have a problem in the gene?
Oh, so what does it mean when we say a problem in the gene? That's a good question. So copy number variance means you're missing sections, right? And it includes multiple genes, so like sections that are deleted. When we say mutations or a problem in the gene, inside each gene, I said it's made up of deoxyribonucleic acid, DNA. And DNA is a series of letters. It's a code. And when we're talking about mutations, we're talking about maybe a letter is changed or switched. So it's not like something deleted. It's actually the spelling. So, it's not that a whole sentence is missing, but there's one word that's misspelled. And sometimes it can have exactly the same effect as if you deleted it. It knocks out the function. But we classify these as copy number. It's a problem with the number of copies, or sequence level, so something with the spelling of the gene.
So if you have a duplication of that 11.2.
1.1.
Sorry, 1.1.2 to the 1.3.1, do you automatically have a mutation in that GABRB3 or not?
If you have a deletion, you mean? Or you said, okay, if you have a duplication, no, not at all. What you have is the gene is probably intact. There's no reason to suspect there's a spelling error, but you have too many copies of it. You're only supposed to have 2 copies of it. If you have 4 copies.
A mutation of that, not necessarily an extra copy of it.
Right, so this just means that even in people who don't have Dup15, these are individuals who have epilepsy, sometimes they find spelling changes, mutations, just in that one gene. That's not the problem in Dup15. The problem in Dup15 is the entire gene is producing too much of what it's supposed to produce.
Just a quick question. So you can't really fix mutations or anything, but say if you were to use a technology like CRISPR, could you go in and just take out like a mutation, say you have too many copies, I just want to cut the copies so that you're not messing with the gene? Would that help?
That's a great question. So CRISPR, you may have been reading about this new technology that allows us to edit genes. There's still a lot of work to be done with CRISPR. So what they're using CRISPR for now, and probably the first breakthroughs using that technology, is going to be at the level of some of these little mutations or variants in single genes, where they're actually editing it and then maybe inserting a gene that works more functionally. In Dup15, because we're talking about dozens and dozens of genes, and it's extra, I don't think that's not going to be our technology. But let me give you another example about how treatment might work without talking about gene editing or something at the gene level. If we find out, for example, this is a gene that's important in epilepsy. What does that gene do normally? What is it typically supposed to do? What pathway in the brain is involved, and how does it provoke seizures when it goes awry? And then you tailor pharmaceuticals or drugs to specifically try to target what's going awry with that. A classic example is Fragile X syndrome, is another relatively common cause of intellectual disability and autism. Fragile X is caused by one missing protein. And so there are actually human clinical trials of drugs to try to replace the function of that one protein in the brain that's supposed to control transmission of signaling between brain cells. And they're very close. I mean, they actually have animal models of Fragile X where they have replaced the function and have reversed neurological symptoms in the Fragile X animals. And then they were able to move on to human clinical trials. So I think that's a much more realistic way that we're going to get there, is probably through understanding what these genes do and then trying to intervene at the pathway level rather than trying to go back in and kind of delete whole sections of genes that are there. Okay, so just quickly here, this is a chart that's showing you how many copies of the genes and the types of variations that we might see. So again, that's chromosome 15 up there. We've blown out the section of interest, and then the yellow section is the Prader-Willi-Angelman region, right there in yellow. Often with the supernumerary, or extra chromosome, isodicentric, let's call it like a million different things, what we used to call inverted dupes, what you have is not only extra of the Prader-Willi-Angelman region, but all the way up until the P-arm, right to that satellite region. And it ends up being 4 copies, because you got your 2 regular 15s and now you got this extra copy. Sometimes you'll have people who don't have 4 copies, they'll have 1 extra copy in their extra chromosome, and it might be base pair, breakpoint 1 to 3, and there's 3 copies and extends a little into the P. Sometimes it's just a breakpoint 2 to 3, and again, these can be interstitials or supernumerary. And then sometimes, as I mentioned before, you have duplications, 3 copies instead of 2, that are outside the Prader-Willi-Angelman region. It's a Dup15, but it's not Dup15Q syndrome. This is a breakpoint 1, breakpoint 2 duplication. And likewise, on the other side, there's 2 different kinds of duplications you can have there that might cause some symptoms, but because they don't include the Prader-Willi-Angelman syndrome, it's not this syndrome. And we're not just trying to exclude people. We'd love to be inclusive, but if we want to be scientific about it, and we want to look at our genes of interest, we're not doing anybody a favor focusing research on our genes of interest in the Prader-Willi region, knowing that these individuals do not even have a duplication of that. They have a different disorder. They need to be looking elsewhere, not within our region. Again, this extra region you can find sometimes. Sometimes you'll find also kind of larger duplications. So if you, the party line is if you hear somebody talking that they have a Dup15Q, one of my first questions as a genetic counselor is, show me your lab report. Are we talking about our Dup15Q with the Prader-Willi-Angelman region or not? Are you talking about a duplication in front of the region, behind the region, or including the region? Edge duplications and core duplications. So there are these dupes that don't involve the region. So they're implicated in a number of different things. They can sometimes cause ID, sometimes schizophrenia, autism, ADHD. They're known, certainly the deletions, and are known to be pathogenic, meaning that they cause problems. And the duplications, not really quite sure the full extent of what they're causing. Same thing on the front end, those little duplications that you can find right before the Prader-Willi-Angelman critical region. We're not really sure what they do. We think they might be associated with ADHD, ASD, maybe some speech-language disorders, but it's really a variant of unknown significance. So finally, your lab report is key, as I mentioned. You're all from different parts of the country and the world. And, you know, this is the last thing I'll say as a genetic counselor. I hear sometimes from parents that, you know, the day they found out that their child had Dup15Q syndrome was the day they were also sitting in front of a geneticist or a genetic counselor trying to learn about the genetics. You think you're going to remember any of that? Absolutely not. And then they never go back. And so, I mean, I would always say, I would let the dust settle into your new reality, your lives, your therapies, and when things are going well, go back and revisit. Any genetic counselor is going to be happy to see you again, take that lab report, and maybe when you're not completely shell-shocked at the moment of diagnosis, which is when a lot of this explanation happens, no wonder people don't remember. And for many genetic disorders, it doesn't matter because it's really easy genetics. It's missing, parents don't have it, you're done. This is a really complicated region. I would encourage you to go back and meet with a genetic counselor.
Because I went twice to my dentist and I didn't get what you're offering here.
Oh, well, thank you, but you know, I get genetic counselors calling me because it's my area of expertise. Don't ask me too many hard questions about cancer genetics. I would do a foul-up job. I know nothing about cancer genetics, but so you know, everybody's got their expertise. But if you're interested and you haven't been to a genetic counselor, if you go to the National Society of Genetic Counselors, they have a Find a Genetic Counselor tool where you can put in your zip code, and then you can sort it by specialty. And what you want to look for is a pediatric genetic counselor, even if your son or daughter or self or an adult, primarily because this is a pediatric condition and they would be the ones to know more about it. Just want to acknowledge my colleagues out at Geisinger. We have one of the Dup15 clinics, and from that, we will go on, and we do have to clear out of this room in about 40 minutes or so, but we are going to be, Carolyn and I, maybe sitting up here, and kind of, we've actually been brainstorming, and we've got those of you who sent reports, we've already kind of written up some things to give you, and we'll go through this quickly. If you did not send Vanessa and the Dup15 Alliance your report, or give your report to us, but you have your report with us and you want to go over, you can certainly share it with us now. So, any other questions? Yeah.
My question is, when you have those edge dupes, like, along with, like, do they, like, compound those extra little symptoms, or is that just, like, by this point it doesn't really matter, that you go through, you know, 1.3.3, then you go to 1.1.1?
So that's a really great question, and of course it's one that intrigues us, like, you know, what does it mean? What we do know, and this is work that's been done on a number of people over the years, is whether you have within that, you know, usual region, whether it's a little bit smaller or a little bit larger, it's not really predictive of anything added. Now, that being said, there have been found in a small number of kids very, very large duplications. And for those, yes, they may have a much more severe presentation. But if you're in the usual bounds of between that 1 and 5, you know, you're typically going to have a duplication. And yes, if you have 6 copies, let's say, you may have more problems. But most people don't. Usually it's 4 or 3 or whatever. So we don't think it And who knows if it compounds, or is it just you have such a big impact from the Prader-Willi-Angelman region that even if it did a little more ADHD, are you going to see it that much? Mhm. So I think the PWACR, that's our big region that's of big impact.
This might be a little bit beyond what you're here for, but could you tell us, for folks like me with an older daughter and folks with younger kids, where do you see things going in the next 5, 10, 15 years? What are your dreams? What do you hope for older folks and younger folks?
Yeah, I have dreams too. I work with a lot of different groups. I always say, you know, Dupe 15 is in my heart, but I work with many groups. And I would say that in the next 10 to 15 years, we're seeing a lot of advances in genomic medicine. And if you had asked me 15 years ago that I would be actively involved in clinical trials of drugs to reverse neurological symptoms in adults with Fragile X, I would have said, oh, come on. I would never have believed it. So we have advanced to such a point that I am extremely hopeful, and I have no idea what will happen. And one of the most important things really are organizations like this, the Dupe 15 Alliance, because it pulls the scientists together who all have an interest around this. And you know, when we started this, we had a little fledgling group of, you know, geneticists or whatever. Now, and then we started, a couple of years ago, the Alliance started having scientific meetings, a half a day, or let's get together at an existing conference. We have a full 2-day conference coming up at UCLA. People are coming in from all over the country, all over the world actually, and really focusing on it. So I don't know what to predict, because I don't have a crystal ball. All I know is that we need to keep fighting for NIH funding and for private foundation funding for research, because phenomenal things are happening, things that we never thought could happen before. We have animal models of human genetic neurodevelopmental disorders. Wow, that's huge. Think of the advances that have been made in cancer over the last 50 years. Cancer was a death sentence 50 years ago. Childhood cancer is mostly curable now at this point. I think we're at the brink of kind of starting that with neurodevelopmental disorders. And I don't think I'd be accused of being too optimistic. I've seen it. We already have a condition called tuberous sclerosis, which is characterized by large benign tumors that grow in the brain, the kidneys, and all over the body. And there's an FDA-approved medication since 2012 that shrinks them down to nothing, and now that is given to kids early on diagnosed with that. So there are some precedents, and there's a lot of excitement going, but we have to keep supporting the research, and we have to keep the groups together and build this, because if we find treatments and everybody's scattered and we have no central organization to say, here's a clinical trial, we need to enroll people, et cetera, we'll never get anywhere. So the fact that this group is growing and is so organized is tremendous.
I just have one comment. It really hit me what you said about in that duplication region, in that Prader-Willi region, that those cells aren't necessarily mutated, they're just duplicated. Could you just briefly explain why is it extra material bad?
Oh, so why is extra material bad? For a lot of genes, extra material or missing material doesn't really matter. They're not what we call dosage sensitive. There are certain, and not every gene in that region is going to matter. It doesn't matter. But there are certain genes throughout the genome that are what we call dosage sensitive. You have to have the exact amount. If you have too much or you have too little. And if you have too little in some disorders, it gives you kind of an opposite symptom cluster from when you have too much. But then there are disorders where it doesn't matter. If you have too much or too little, it's going to give you the same problem. We don't know quite exactly what's going on, but we do know clearly UBE3A is dosage sensitive. You get Angelman or Prader-Willi if you're missing it, and now you get a different phenotype or group of characteristics for it. So yeah, it's not every gene, but there are some genes that are dosage sensitive, and they're the ones we want to be careful, and we're the ones we really want to focus on. And so if there's too much being produced, what does that cause, and maybe how can we intervene, maybe not at a gene level, or maybe, but even just secondarily to block that extra product that's there. Come on up, because you're going to come up anyway, and you might as well take the mic.
I'm just dying to be on the stage.
Oh, I know.
Hi,
You
everybody.
have to pull that down, you're shorter
I
than I am.
know. I used to get to do this all the time. So to answer that question a little bit too, which actually feeds into the GABA beta 3 thing. So GABA beta 3, which was the gene she showed that was mutated, is actually one of the dosage sensitive genes. And they've shown in epilepsy models that how that gene, genes encode proteins. And the protein that makes actually has to work in a group of with four other proteins to build something that's the GABA receptor that sits on the cell. And a lot of the epilepsy drugs work on GABA receptors. And so that particular one, if you make too much GABA beta 3, it actually floods the system and that whole receptor doesn't form. So when they were trying to work in cell systems and they would make too much GABA beta 3, what they found out was actually they ended up making not enough. And so they couldn't get it out onto the cell surface because they were making too much of one. And so you have to make it so that you make all five of them in a balanced amount, or else you'd end up with not enough. And so that's one of the particular dosage sensitive genes. The other thing I was going to mention is that there's a disease called Pelizaeus-Merzbacher disease, which has nothing to do with DUP15 other than it's caused by a duplicated gene. And there's a clinical trial ongoing right now to shut down that gene using a targeted approach to just literally go after the gene. And it's because they know what the gene is, right? And it's having some effect. And it's a brain disease, pure brain disease. And I would never have guessed. This is one of those ones where the kids have problems from the get-go. And I would never have guessed you could do something for PMD. And the kids are improving. And so it's one of those ones where it's like, it adds hope. So if we know GABA beta 3 is one of our epilepsy genes and we could do something about it, shut it down using either a drug that could change the levels of one or the other, we could know that it's that one that we need to target for, therapies, either drug-based therapies or potentially through one of these gene, regulatory pathways. And not really gene therapy, because that's not what this is. It's a different way of doing it, but it still works. It's going to be deadly expensive, but it's going to, if it changes how kids grow and develop, it ends up in the long term being much less expensive. But anyway, there are lots of things to be hopeful about.
It is, and we've both been around like a long time, and I'm always an optimist, but never in my wildest dreams did I see where we would be going, as I say, with clinical drug trials. I've always got my ear out for all the different syndromes. DUP15 is my favorite. You're not supposed to have favorites. I do cheat on you sometimes with other syndromes, but I've always got my ears out. It's like, oh my gosh, could this really be happening? And to me, having the Human Genome Project leading to molecular mechanisms and the ability to create animal models of human genetic disease, that was a huge breakthrough for all genetic disorders, because now we have something we can really work with and understand. You asked that push button question. We obviously get very excited about it, and maybe not in the next decade, but certainly 20 years will look a lot different than it is now. Maybe not in time for your kids. Maybe it's something that has to be early on, but I certainly wouldn't count it out. In Fragile X, some of the mice where they reverse the neurological symptoms, they weren't able to do this in the humans. It's more complicated, but at least we've got a mouse model. Those mice were elderly mice, and they were able to restore neurologic function. So it's not just like baby mice, let's prevent. In some of these animal models, there's tremendous hope that they've actually been taken a mouse that has the equivalent of like a lifelong chronic disability and reverse the findings. So I think we're at infancy stages, but it's important to keep your eye on the research. Thanks.